Hello friends, good afternoon. Welcome to Sam Biology class. Today we shall discuss about the ultrastructure of eukaryotic chromosomes. Before that, I request all of my friends, please subscribe my YouTube channel Sam Biology class. Chromosomes are rod-shaped and self-replicating organized mass of DNA. They are visible only during mitosis and meiosis. During interphase, chromosomes are not visible because they unfold and uncoils into a diffuse network of fine threads called chromatids. But when the cell enters the M phase, the chromatin threads condense or folds or package into rod-shaped chromosomes. As a result, they become visible. So when a cell progresses from M phase to interphase, chromosomes are decondensed, but when it progresses from interphase to M phase, chromatin threads are condensed or packaged into chromosomes. So friends, today we shall discuss as to how chromatin threads, which are long molecules of DNA double helix, are condensed into rod-shaped structures called chromosomes. All eukaryotic organisms have special ways of packaging the DNA into chromosomes. For example, the 48 million nucleotide pairs of human chromosome 22, when laid out as a single long perfect double helix, it will extend for 1.5 cm if stretched out end to end. But during mitosis, chromosome 22 is only 2 micrometer long. This represents an end-to-end compaction ratio of over 7,000-fold. This remarkable compression of chromatin threads into rod-shaped chromosomes is performed with the help of proteins which successively folds and coils DNA into higher and higher level of organization. Today we shall discuss about models which explain the packaging of DNA into chromosomes in eukaryotes. One of the model is folded fiber model, which was proposed by E. J. Duprat in 1965. According to this model, chromosomes are made up of chromatin fibers, which are about 230 to 300 angstrom in diameter. Each chromatin fiber is produced by coiling of a single long DNA double helix, which replicate during interphase, producing two sister chromatin fibers. As 0.3% of DNA remain unreplicated during S phase, the two sister chromatin fibers remain attached at the centromeric region until their replication during the zygote substage of meiosis I. According to this model, it is because of folding, the two sister chromatin fibers become short and thick. As a result, two sister chromatids are formed. Another model that explains the packaging of DNA into chromosome is Nucleosome solenoid model. It was proposed by Kornberg and Thomas in 1974. This is the widely accepted model. The proteins that bind to the DNA to form eukaryotic chromosome is traditionally divided into two classes. Number one is histone and number two non-histone chromosomal proteins. The complex of both these proteins with the nuclear DNA of eukaryotic cells is called as chromatin. When interphase nuclei are broken open very gently and their contents are examined under electron microscope, most of the chromatin appear to be in the form of a fiber with a diameter of about 30 nanometer. When subjected to partial unfolding, It can be seen as a series of bits on a string under the electron microscope. The string is the DNA and the bits are nucleosome core particles which consist of a histone octamer around which the DNA is coiled around. This is the form of a chromatin which looks like a series of beads on a string under the electron microscope. The beads are the core particles and the string is the DNA. So in this chromatin fiber, the DNA is coiled around a number of nucleosome core particles. The stretch of DNA which is present between core particles is called as linker DNA. The length of the linker DNA varies from few nucleotide pairs to about 50 nucleotide pairs. The structural organization of nucleosomes can be determined after isolating them from the unfolded chromatin by nuclease digestion. The enzyme nuclease will break down the linker DNA, thereby releasing nucleosome core particles which are about 11 nanometer in diameter. The core particles can be dissociated into an histone octamer and a DNA double helix which is 147 nucleotide pairs long. The histone octamer can be further dissociated into individual constituents which include two molecules each of histone H2A, H2B, H3 and H4. This is a nucleosome core particle which consists of a histone octamer and a double-stranded DNA which is 147 nucleotide pairs long. The histone octamer is a disc-shaped structure. It consists of a complex of 8 histone proteins which include 2 copies each of H2A, H2B, H3 and H4. The double-stranded DNA is tightly wrapped around the histone core in a left-handed coil of 1.7 tons. Histones are small basic proteins having 102 to 135 amino acids. All of them share a common structural motif known as histone fold. Because of the presence of high proportion of basic amino acids, histones are positively charged, as a result of which they easily bind to the negatively charged DNA. In addition to core histones, nucleosomes are also associated with histone H1, which is known as linker histones. A single molecule of H1 is associated with each nucleosome, contacting both DNA and protein, changing the path of DNA as it exits from the nucleosome. By changing the path of DNA, it is thought to help in the packaging of DNA. As already mentioned, the length of the linker DNA varies from few nucleotide pairs to about 50 nucleotide pairs, and the double-stranded DNA, which is tightly wrapped around the histone core, is 147 nucleotide pairs long. So, on an average, nucleosomes are repeated at an interval of 200 nucleotide pairs. It is by the formation of nucleosomes a double-stranded DNA molecule is converted into a chromatin fiber which is about 1/3 of its initial length. So histones are responsible for the first and most basic level of chromosome packaging. Due to the formation of nucleosomes, the long DNA double helix becomes short and thick. This resulting chromatin fiber appears as a series of beads on a string under the electron microscope. This chromatin fiber, which is 11 nanometer thick, is also associated with non-histone chromosomal proteins which play specific role in gene regulation. In the next step, the 11 nanometer thick chromatin fiber is further coiled to form a specific structure called solenoid. The solenoid structure is 30 nanometer thick. It is a chromatin fiber of packed nucleosomes. The solenoid structure has 6 to 7 nucleosomes per turn. In the next step, the solenoid is further coiled to form the super solenoid, which is 300 nanometer thick. In the mitotic chromosome, the super solenoid is further condensed to produce the final shape and dimension of metaphase or anaphase chromosome. In the metaphase chromosome, two daughter DNA molecules are separately folded to produce two sister chromosomes called as sister chromatids that remain attached at the centromere. So the packaging of DNA into chromosomes is a wonderful process in which proteins successively coil and force DNA into higher and higher level of organizations. As a result, rod-shaped chromosomes are formed. So friends, this is all about the ultrastructure of eukaryotic chromosome. For more update, please subscribe my YouTube channel, Sam Biology Class.